So let me introduce myself. As Carmen said, I'm her colleague. I work for Thermo Fisher Scientific. It is a global company, provides different scientific instrumentation, reagents, consumables. And so I'll be speaking today about, I guess, a slightly different perspective to genomic data. I want you to imagine a scenario, right? A very realistic one. Your favorite social media platform gets hacked. Quite not a far-fetched situation, right? What do you do apart from getting angry and maybe ban that social media platform? Well, you delete your maybe banking details, change them, change your password, right? Move on with your life. Angry tweet or something. What happens if your genomic information gets leaked? You can't change your DNA, at least not yet. Nor can your children, your grandchildren, their future children, any of your relatives really can't change their DNA, right? Next thing you know, your health insurance premiums go up. Maybe because, I don't know, you're likely to develop Alzheimer's in the next 10 years. Your mortgage application gets denied for the same reason, or your grandchildren's application gets denied. That can be pretty scary. I don't mean to scare you just yet, maybe a little bit. But that's just one of the challenges, the unique challenges of working with genomic data, especially as it transitions from research into the clinical space. Oh, there we go. Quick disclaimer. So we've seen a huge explosion of data in the biotech industry over the last 10 years, right? And the culprit is the new advances in biosciences and technology. So I'm going to talk today a little bit, maybe refresh your knowledge about what DNA sequencing is, to build on Dr. Fatima's talk, and also present you with some maybe less obvious challenges that come with it. So for those that just joined, or maybe those who had a little bit more than just coffee during lunch break, DNA sequencing, what it means, right? We all have an idea of what DNA is. It's some sort of molecule, has our genetic information, the ultimate instruction manual to a cell about how to be that cell. And it's made of the 4 building blocks, the A, T, C, and G. And so reading that sequence is basically what DNA sequencing is. Why do it? Well, it's interesting to research it. Sure, we can maybe predict certain things. But as we're moving toward the clinical space and actually applying that to real life, to real patients, we can do extraordinary things with it. We can prevent, monitor, diagnose, treat cancer. We can use it for relevant today epidemiological studies and diagnose infectious diseases. Moreover, look at their evolution over time. We can use it for reproductive health. Take 2 people. What are the risks if you have a child? Moreover, in animal health, what are the biomarkers that makes that particular cow's milk the most delicious, or that tomato the most transportable? Forensics, drug discovery. It is already here and we're living through it. How did we get here? It all started in 1870 with the discovery of DNA. Then just over 100 years later, Frederick Sanger and Walter Gilbert identified the first way to sequence DNA, and that was called Sanger sequencing. Walter didn't get the name there. So that was the very first way to sequence DNA, and to this day it is considered to be the gold standard. However, later in 2005, the development of next generation sequencing, so the generation after, led to a significant improvement in speed, in volume, right? If we had first human genome sequenced in 2001, it was drafted for $3 billion, 2.7, then with the development of NGS, later in 2008, we could sequence it for much less, and today for less than $1,000. This graph in genomics is used and abused every time, but it does signify how fast we've gotten here, really. It is extraordinary. Moore's law, technology that keeps up with Moore's law is considered remarkable. It's great. This outpaces it like nothing. So what is NGS, next generation sequencing? It's simple. If Sanger looked at one fragment at a time, usually we'd look at one or two or a few genes at a time. Next generation sequencing lets you to look at millions of fragments at the same time. So it is deep, high throughput, and parallel DNA sequencing technique. And boy, it produces a lot of data, up to 40 exabytes of data. We've heard a lot about petabytes, exabytes, all of that. I had to look it up. What is an exabyte? How can I actually compare it? Well, in fact, 40 exabytes is 8 times the size of internet today. What about, say, a gigabyte versus an exabyte? If a gigabyte was a grain of sand, an exabyte is a tennis ball. Imagine up to 40 of those. That's the prediction for 2025 of what genomics is going to be like. And it creates a lot of challenges, right? The obvious one, computing and storage. And as we move from research more into industry and the clinical space especially, and we actually embrace that technology for the good of the patients, storage and computing that can be kind of resolved with utilizing the cloud has to be considered with a grain of salt, because security is a very big issue when we're dealing with patient information. That security, as you saw in the beginning of the talk, can have huge implications if not embraced and respected and considered, right? That security concern is also what is inhibiting data sharing a little bit. As research is pushing for more and more data sharing, and that 40 exabytes, even just interrogating a small piece, would be great for our common good, right? But that data sharing is also inhibited by an aspect that's not commonly looked at, the social aspect, right? If we have so many different players from so many different sectors, the industry, think of your R&D scientist in a biopharma. He has a very different motivation, right? Different goals, very different reward system. They're not as inclined to share the data, right? They're going to lose competitive advantage. Your research scientist, very different goals, reward system, motivation, norms. The norm is to share. Within research, the norm is to gain recognition by publishing. Be there first. What about clinical space? Again, very different. You're a clinician. The goal is to take care of you, the patient, right? Sure, clinical science, all of that is important as well. But getting everyone together is quite a challenge. And building a system that does recognize the differences and does provide a new, I guess, reward system to share that data. And then, of course, the legal side, the least fun. But if you think about international data sharing, right, it's different jurisdictions, different countries. Just the sharing between the industry and research can take months and months to get the legal contract set up. We know that very well. And then the privacy debates, right? We've talked about the social aspect when it comes to the person who has that data and shares it with the research community or the clinician community. But what about us, the patients, right? Do you want to have your data shared? What kind of consent did you sign? Like, that is also important to consider when it comes to public's trust. As we want to get more and more data from more and more people, right, to have a representative, unbiased data set, we would have to tap into the more, I guess, skeptical ones who aren't as willing to share their genomic information. How do we convince them that it's okay? Multiple surveys have shown that actually we are all more likely to share our data with the clinician. Second likely with the researcher. Very unlikely with the industry scientist, right? Makes sense. We've been accustomed to the news and we've been accustomed to sharing our personal information with the clinicians. So taking that into account is also important. And unfortunately, we talk about big data, it's rarely that we take into account the human side of things, right? Then data integration, the purely big data ultimate challenge, right? You have heterogeneous data all over of different formats from different analysis pipelines with all the additional things that we want. We don't just want the DNA. We want to know what's the fat percentage of that particular cow, what's the associated clinical status of that person, what's the height, the BMI, whatever we want to look at, we want all of that information too, right? To find that pattern. And yet it's very difficult. There are many gaps, there are many differences in formats and systems, and navigating that can be a nightmare. And the last one, that is my personal favorite, is the data interpretation. Interpreting that data, unfortunately, cannot be fully done by algorithms yet. So you need a lot of people with expertise that can literally find a genomic needle in a haystack, because you have all of that data overload, all of that, 98% of our genomes considered to be garbage, probably not realistically so, but for now, we just don't know a lot of it. And looking through all of that and finding maybe this mutation caused this cancer can be really challenging and requires a lot of expertise and time and sifting through publications. And one person will say that this is a likely pathogenic mutation, and another person will be, no, this is an unknown. I can't be confident of. And that is the difference in interpretation, just because there's still lack of data out there, right? That creates another challenge. However, all of these challenges, they are out there. And smart, innovative scientists are working to toil away at them, right? The biology field has evolved into a big data domain, and it's constantly growing. Even though these challenges exist, they will be overcome, but we do need to keep in mind the social aspects of it and the privacy aspects of it. That's it from me. Thank you. Any questions?
As per usual, great talk. Thank you very much, Alex. Thank you. So we will give only one question because of the time constraints and because we are going to the break after this.
Hi, Alex. Alexandra, sorry. Fascinating talk. My question is there a way to mask or de-identify or de-anonymize DNA? Because that would solve the problem.
This is a fantastic question. I actually had it in my talk. I decided to skip it for the benefit of time. Great question. So yes, that is one of the ways to share data by de-identifying the people. But if you think of the nature of your DNA sequence, if you have enough, it has unique identifiers within it, right? It's like fingerprints in a way. Yeah. And so with enough information out there, and studies have shown, you can actually re-identify that person. So a good example is the Golden State Killer. I don't know if you're familiar. It's a California serial killer that has been caught by law enforcement based on a match of his DNA from a crime scene to his distant relative's DNA that was uploaded to Ancestry website. Now the access to that, it was public access. Great law enforcement used it, but think just of some potential implications of doing that without the good intentions, right?
But what if you encrypt like the same way that we encrypt information? So it's very difficult to de-encrypt until you have the code. I mean, I'm just talking from my point of view of ignorance of DNA.
Yeah, absolutely. There are encryption techniques for it as well. So the cloud, I guess, storage and computing on the cloud is currently embraced by many, but still have hesitations around it, purely because the legal implications. So if a clinical scientist is working with the data on the cloud and something goes wrong, and Amazon Web Services say, oh, I'm sorry, we are only 99.9999% secure, then the legal implications fall on the scientist, not on the third party. So that sure, encryption is a good way to use it, but there are just cultural hesitations around in the field at the moment.